All right, guys, I just wrote this title right now, so it's a very lame title of the talk, very generic. But in the next half an hour or 40 minutes, I'll take you through very quickly over a few works from our lab, primarily in cancer. And it's important to say that we are not only, we started our lab working on single cell genomics, and we wrote a lot of algorithms to scale up single cell genomics. But over the time, we also pivoted a little bit to cancer. This was always an interest for us, so we pivoted a little bit to cancer genomics and try and see that how we can define, and this is how I like to identify our works. We wanted to provide computationally tractable definitions to some oncology grand challenges, like detection of cancer from blood. Just process that what I'm saying. We wanted to define the problems in a manner so that they're computationally tractable, and then solve it with some methods. So I don't want to glorify the methods that we built right here, but I want to glorify the fact that many of the major challenges in cancer can be tackled by computer scientists like me or my other team members who never studied a single course in biology in life. Okay, but still we can contribute. Right. As we know, the human body has about 40 trillion cells, and each cell has one copy of DNA and several RNA molecules floating around, about 20,000 different RNA molecules, right? And you can really be unlucky to get a mutation at one particular base, and that's good enough to make you sick enough to die, right? So that's cancer for you. And cancer is uncontrolled growth of the cells, because you hijack the growth mechanism, the growth signaling, in a manner which becomes uncontrolled, and the cells proliferate and expand much, much faster as compared to the other cells in the body, the normal cells in the body. Okay, so I'll be primarily talking about two very important areas of our research, and then towards the end there is another topic. If I feel good, I'll probably, and that's a little blue sky. So there are three stories that we'll be sharing here. The first two can be described through this simple slide. There are two major problems in the world of cancer. The first is diagnosis of the cancer, and the second is treatment of the cancer. When we talk about diagnosis, there are different stages of the cancer, so diagnosis mechanisms are very different. But the question that we are trying to answer is how to detect cancer from blood, which is much easier, because it's just easy, right? It's just like you do a diabetes test, you want to do a blood test for cancer detection. And how wonderful would that be if you can get it at a cost which is low, right? So that's the question. And within that, we'll talk about detection of circulating tumor cells here. On the treatment side, we'll talk about the targeted therapies, and under that, AI-guided therapies, primarily talking about how we can use chemoinformatics and bioinformatics in conjunction to predict how a drug would act on a person given his or her cancer profile in terms of transcriptomics. The first part talks about the microfluidics, single cells, transcriptomics, and artificial intelligence for CTC detection. A lot of things. It's a very interdisciplinary work. It's not a work of our team alone. There are people from 4 different continents who actually contributed. Towards the end of it, I'll introduce them too. But it was led by our group, and I'll tell you who is the main person behind it later on. But if you look at the cancer detection space, so at an early stage, in a healthy individual, it's not really easy to detect cancer. I mean, if somebody is already healthy, just by looking at the DNA, you can probably have some well-known mutations which relate one, predispose one person to the risk of cancer. One good example is Angelina Jolie, who underwent surgery because she was at a risk of breast cancer. But that's not quite a market here. So not that there are many such markers, biomarkers, which correctly tell you that there is a significant risk. So leave that aside. But when it comes to early detection of the cancer, there are few solutions. I don't know whether you have heard of GRAIL. So GRAIL is a big company. They have a ctDNA-based method for cancer detection. They basically profile the methylation patterns of the circulating tumor DNA or cell-free DNA. That's one modality. A lot of people actually started looking at the circulating tumor cells also, but that didn't really fly well because circulating tumor cells are extremely rare in the blood. So, and also it requires certain kinds of equipments and instrumentation and microfluidics to be able to catch them. And therefore, it was not found to be viable, right? And then if you look at the follow-up phases, there the cancer is little more detectable and the damage has already happened, right? So there you can find out the mass through scans or other intrusive methods. Okay. Okay. Now, if you look at since we are talking primarily about the liquid biopsy, there are the following modalities of the liquid biopsy methods. So, have you heard of circulating tumor DNA? You have. Circulating tumor DNA are the DNA being released by cells, cancer cells that are dying. We always have cells dying in our body and they release DNAs into the bloodstream. Cancer cells do the same thing and you can trace them back in the blood. And if you can, then you can probably recognize that this person has cancer. And their admixture, the tumor cell circulating tumor DNA fragments would have some kind of impact on the signal of cell-free DNA in general, which can be detected, but it is actually very difficult to segregate between the two. You can only look for the canonical mutations, or you can look at some patterns in methylation, etc. So in the end of the day, you need artificial intelligence to dissect whether it is coming from a normal person or a cancer. Now, circulating tumor DNA is that. And then comes the circulating tumor cells. By the way, circulating tumor DNA have a lot of use in clinic, because when you know somebody has cancer, you can keep monitoring the person with their varying level with the progression of the disease and the treatment. You can measure the level of the circulating tumor DNA in the blood, and that's well practiced in the real world, right? So circulating tumor DNA is really a very well used biomarker for cancer detection and treatment monitoring. So I don't mean to demean that. There are two other modalities. The first thing is circulating tumor cells. Circulating tumor cell is another very interesting biomarker because they are de facto cancer cells floating around in the body, right? They come from the tumors. So actually, just because you cannot detect cancer because circulating tumor cells are extremely rare does not reduce the importance of the circulating tumor cells, studying circulating tumor cells. Because imagine that if you can catch all the circulating tumor cells from the blood of all different phenotypes, not only the ones which are epithelial in nature, but everything, then you can learn so much about the disease biology, right? Because you cannot, so circulating tumor cells, given that they're always floating inside your blood, that gives you a temporal snapshot time to time about how your cancer and tumor is changing and how it is reacting to the drug. So it is extremely valuable, okay? When you catch hold of circulating tumor cells, you have the whole cell with you. You can study the DNA or the RNA, it's up to you. You can study whatever you want, but catching them is not that easy, given that in 500,000 WBCs, you will have one circulating tumor cell. And that too, in many cancers, you will not find. The other interesting thing we came across back in 2013, 2014, when I was doing postdoc in Genome Institute of Singapore, that tumor-educated platelets, or TEPs, are the normal body platelets that have been shown to undergo significant change in its transcriptome, right? That means platelets are basically garbage cans. They are not even cells. They are cellular fragments. They don't have their own personal nucleic acids. All they have is nucleic acid from the other cells. I don't know where they collect it from, but they exchange nucleic acid. So at the end of the day, if you tear the platelet, you'll see some nucleic acid, you'll see gene expression, you'll see everything. You'll not see a DNA, but you'll find gene expression. And you can profile the gene expression in the platelets. That's very common. You have plenty of platelets. And then it has been shown by Best and colleagues back in 2014 in a cancer cell paper that platelets undergo significant change when it comes to cancer. And they applied artificial intelligence in a way, a simple machine learning method, I would say, not deep learning or anything. They use simple machine learning method with a great confidence, predict this platelet is coming from cancer patient and this is coming from normal ones. So it was very exciting for me, because when I thought about implementing back in 2014, when I was doing postdoc in Genome Institute of Singapore, I thought, how can we perhaps create liquid biopsy in India, because it was not there at that time. People were basically borrowing technologies from outside countries, right? So I asked that, can I use platelets? Because platelets, just imagine, I never studied biology, but it's not that difficult to see that if platelets are in the body and platelets are undergoing so much of changes, can you pick up just a few genes in the platelets? That's a feature selection problem. And if you can pick up a few genes from the platelets that confidently predict the existence of cancer in your body, then that will be wonderful. And if you have only a few genes, you don't even need to do genome sequencing or transcriptomics. You can simply do a PCR, right? On a single run, you can get like 12 genes expression, all right? That's pretty interesting. So we did exactly that, and I'll show that result later on. So basically, so to say that these are the three top modalities that excite us. There are so many other things like extracellular vesicles and all of that. We are not working on that. Doesn't mean that these are not important. These are very important. It's just that we happen to work on these two modalities, circulating tumor cells and platelets. Platelets were more from the implementation point of view, like how I implement liquid biopsy for Indian people, because they cannot afford microfluidics. They cannot afford anything to detect circulating tumor cells. And circulating tumor cell was from the point of view that if I can catch all kinds of circulating tumor cells, not only the ones that show EpCAM or epithelial markers on the top, not a marker-based selection, if I can identify circulating tumor cells of all kinds and phenotypes, then that will be awesome for me to study biology of the cancer and as it goes. Okay, so that's the idea, guys. I don't know. So this is how a single circulating tumor cell looks like in this big maze of so many cubicles. It's like 500,000 to 1 ratio, right? So they are extremely rare. We thankfully had a lot of ideas about how to deal with single cells before we actually embarked on circulating tumor cells. You cannot build a castle in a day, right? So we initially started studying how to detect rare cells in the patient, in blood, right? Like dendritic cells are also rare. Megakaryocytes in the blood are also rare. Monocytes are not. So the question, so if you see our papers, like FIRE is Nature Communication back in 2018, we came up with a big data algorithm to detect single cells, which are very rare in blood or anywhere, from a big population of cells. Similar kind of technologies, we just changed it a little bit. We use different kind of mathematics, but the idea is how to detect rare phenotypes accurately. So I think that sets the stage to take you through the first invention, which is a pipeline that combines microfluidics with size-based selection and single-cell sequencing, followed by artificial intelligence-based classification of the tumor cells versus the normal. Now, it is a collaborative work between many people. I would introduce them later. But you basically start with blood, and then you use this, which is a spiral chip. And this spiral chip is a microfluidic device which does a simple selection of big cells and small cells. And the big cells may be circulating tumor cells. So from one outlet, you get cells which are bigger in size and weigh heavier. And then from the other outlet, you get cells which weigh less. So the common assumption here is that the circulating tumor cells would have higher weight because they are cancer cells. They're big. If that is not true, then it will fail perhaps. But then we use microfluidics. There is a system, I forgot, Polaris, right? Polaris is a fluidigm system in which you can incubate a single cell. They did some further, depletion for the CD45 plus. Basically, we did a size-based segregation, which is pretty good, right? Because you are not picking up cells with a particular marker. You know why? Because in cancer, you have phenomena such as epithelial to mesenchymal transition. You would expect some epithelial cells, when they are becoming metastatic, they're actually losing their epithelial properties, right? And they are becoming mesenchymal, which are more like spindle-shaped cells, and they are rubbing off each other, and they are moving into the bloodstream. So that's the situation where they lose a lot of their epithelial-like properties. So that's a concept. I'm not sure how true is that, but it's actually true that not all cancer cells display the epithelial markers, which cancer is an epithelial disease, but not that all cancer cells in the blood would show epithelial markers. Then how can you catch circulating tumor cells in a marker agnostic way? So no matter how much of enrichment you do, those are approximate. You would still have admixture of WBCs, which are unwanted, right? So what we did was we did a CD45 depletion, CD45 is negative enrichment to get rid of CD45 plus positive cells to ensure that I have a further enrichment of circulating tumor cells in the population, followed by single cell sequencing and then analysis, which is basically a tool that can predict whether this is a circulating tumor cells or blood cell. So this work was published in 2023, not 2022, in Genome Research. You can check it out. I will share with you too. Unbiased recognition of circulating tumor cells is what we wanted, and that's a multi-step process, which is, I don't know whether, okay. So this is a complicated equation. What it does is it brings the gene expression matrix down to a latent space. So it's called deep dictionary learning. This was done in collaboration with Anshul, who is a triple IDD faculty and a signal processing guy. So he developed, so we kind of collaborated to develop this method, which brings the data in a latent space first, and then does k-means. So it combines the cost of k-means. It does want to do clustering of the cells. At the same time, it wants to do the clustering in a latent space, and both these things are happening together. It mimics deep learning, but it is basically matrix factorization. So these are different layers of, like, these are called dictionaries, basically. These are latent basis vectors in a way, right? So they represent the abstract form of the data, like you do in the SVD, for example. So they are done in multiple layers. You are splitting the matrix in multiple layers, as if matrices are multiplied, and you are trying to reconstruct the original values. And those layers of abstraction are same as deep learning. So the underlying mathematics you can check, but this is called deep dictionary learning. So this is a deep dictionary learning, okay? Then this is just a dimension reduction problem. Yeah. You can do that too. You can do that too. You can do that piecemeal too. It's just a fancy way of doing it together. And that probably works better than doing it piecemeal. So we have tested it briefly. We didn't really share the result of how it compares with, but you were absolutely right. They can be done in sequence rather than being together. All right, so that with I don't know, this is behaving a bit, okay. So that is the clustering. So we get single cells. What we do is, instead of relying on the expression values, we bring them down to the pathway space. Before even applying the mathematical equation that you saw in the previous slide, what we do is we have the gene expression matrix, but the gene expressions are stochastic in nature. So what we also do is we take the gene expression values, and we take some of the important pathways in thousands, right? And then we convert the gene expression values into pathway enrichment scores. And then we apply this method on top of that. And that pathway transformation teases out a lot of unwanted noise in the system. Did you understand? So we demonstrated that earlier in one of the Nature Genetics papers, on colon cancer back in 2017. But just let me progress from here. I think I'd cut it short, just directly go into the result section. Yeah. So, so if you follow this figure, these are basically scatter diagrams, UMAP plots, which are very typical for single cell data presentation. And you have two different kinds of dots that you could say each dot is a single cell. What we did was we constructed two data sets by combining circulating tumor cells from different studies and blood cells from different studies. As simple as that. And you may ask me why. We wanted to make sure that the method teases out the batch effect. In the world of genomics, the biggest monster is batch effect. So we wanted to show that this method that we have built actually teases out the batch effect and gives me the right answer. And if you see these figures, these are the colorful cells are basically representing the circulating tumor cells coming from various studies. And the gray ones are the blood cells. And if you see here carefully, then by Seurat, which is one of the best methods for single cell analysis that we all know who deal with single cell data, it does a fair job here. There is another example in the paper where it does not do well. But here I have just shown the result where it does well. You can check the paper for the other one. But we also do pretty well here. If you see, all the circulating tumor cells are clubbed up together. And that's great. And these are coming from different studies, and there's a clear segregation. Now, once we have that cleanliness in the clusters that we receive, then we can actually characterize, we can look at the markers in the end of the day. So now we are looking at a bunch of cells. So we can take the average values, or we can look at the distribution of different markers that we know. One thing we can also do is pretty interesting. Let me show you one just for the educational purposes. There is something called pseudo infer CNV. How many of you know about it? Infer CNV. From single cell expression data, you can actually infer the structural changes in the cancer genome. Because the genes, the idea is very simple. If you have a genome, and if there is a certain area in the genome which got duplicated or deleted, then the genes that come out of that will also get doubled up or depleted, right? And if you just order the genes as per their genomic coordinates, and you do an averaging out, you do a weighted average of the gene expression, then you can actually come up with this kind of patches. So each column is a cell here, and this dark colors, whether it is brown or blue, would mean that there is a amplification or deletion. That's a wonderful method. When you have a cluster at a cluster level, you can check. By the way, this method doesn't belong to us. This was created back in 2013, I guess. The first tumor single-cell RNA sequencing paper by Bernstein's lab in Harvard. So they invented this method, then it underwent some modifications, but this is a absolutely brilliant way of using gene expression values to infer the big structural changes in the cancer cells. And that is one layer of validation. So the pipeline that we are talking about is an absolutely practical pipeline which takes output from any device where you can have circulating tumor cells and some other cells. Not that, imagine, you are not sure whether the circulating tumor cells are actually de facto cancer cells. They could also be blood cells. You're not sure. Not that all of them are showing EpCAM or whatever. In such situations, you want to have this kind of software which can do a number of analysis to confirm that this cluster is actually circulating tumor cell cluster. And this is what we did. And you can think it's a holy grail of cancer biology. Do you understand? Because if you can study all kinds of circulating tumor cells independent of their markers, the markers that they exhibit, then you can learn so much about the tumor biology, okay, which has so far been very difficult. So this is an example. You remember what I started the talk with? I said our lab gives computationally tractable definitions to oncology grand challenges. This is how I identify ourselves. So nobody has so far defined this as a computational problem. So we provided the framework to the circulating tumor cell recognition as a computational problem. Don't worry too much about the methods. You can apply any decent method that will work. I really don't want to oversell on the methods, but the good part is what we did worked. Okay, so just let's progress. So this is a plot that shows the clusters that we received are pretty homogeneous, like circulating tumor cell clusters are the blue ones. And the other methods, Harmony and FastMNN, they have a little bit of admixture, haywire. Seward does a decent job though, but in a different data, as we said, that it does not do equally well. So one major benefit, as you can imagine, would be, now here comes the clinical understanding, because just outside of this room, I was talking about the importance of knowing the domain. So we thought that what will be the most classical application of this method? The most beautiful application of the method is, can I use this method to determine circulating triple negative breast cancer cells which do not, which are known to not show any markers? So if you are claiming that your method can detect circulating tumor cells without having any specific set of markers expressed on it, then can you detect circulating triple negative breast cancer cells? By the way, triple negative breast cancer is the most aggressive form of breast cancer. And there is very little therapy, very little ways of identifying the cancer in the patient in the first place. So with this method, we collaborated with a hospital in Singapore, and we came up with, we profiled patients of different kinds of cancers, including triple negative breast cancer patients, and we could determine the circulating tumor cells within the same cluster of other circulating tumor cells, and we could thereby correctly identify that the cells are circulating tumor cells from all the patients. So it kind of worked like charm. So that's one good output of this particular method. So this is probably one of the few, or probably the only method that talks about detecting circulating triple negative breast cancer cells, which are known to be notorious, which are known to be not showing up any markers on the surface through AI and microfluidics, right? There is very few methods who do that. Let me just progress from here a little bit. This is the paper that describes all the things that we talked about. And it is a collaboration, as I said, between many countries. So Naveen is a key guy behind this. He helped in creating this data. We created this method. Anshul played a key role in the method development. Sarita, who is our lab member, is now in Cleveland. She led this story for several years, actually, 3-4 years. Briefly, I'll probably talk about the tumor educated platelets, as I began with. So this is another modality of detecting cancer from the blood, but this is much more practical and this is much more inexpensive. The previous one was more to understand how the cancer biology works, and this one is to really bring it to bear to detect cancer in patients in practice at a cost that is affordable in India or any other country for that matter. Okay, so what we did was super simple. It is not as exciting as what you saw in the previous. There's no flashy equation here. It's very simple. There was a study. There was a study published in Cancer Cell, and we took their data. It had platelet gene expression profiles from 5 different cancers and control. And the cancers are glioblastoma, breast cancer, ovarian cancer, hepatobiliary cancer, and prostate cancer, I guess. So all these 5-6 types of cancers and the control. And what all they did was they did a bulk transcriptomics of the platelets from the cancer patients. So they had so many samples, 300, 400 samples, and they applied AI and simple machine learning to predict the existence of cancer. And the prediction accuracy was fantastic. It was like 96%. So the problem was already solved. And it is one of the landmark papers of liquid biopsy research that opened up a new possibility in liquid biopsy-based detection of the cancer. But the problem does not end there, because they did this classification. They reported the classification accuracy with 1,000 genes. So doing the transcriptomics of 1,000 genes is not easy. That would require you to do sequencing after all. The question was how we can bring it down to 10 genes or 11 genes, which can be used on a PC. We can screen on a PCR plate. We can, in fact, if we have a 96 well plate, we can multiplex like 8 patients, right? Or 5, 6 patients, and let's say 12 genes. It's super cheap then. A single PCR plate, it doesn't cost anything. It can happen in a roadside lab. Right? That's exactly what we did. It was just a simple feature selection, which we studied back in our PhD. And here's an application. So we took their data, we applied feature selection to come up. We used a set of feature selection methods to come up with a set of 11 genes, which could actually give us decent accuracy. In that, we used methods because this called eigen sampling that actually inflates the number of samples to help you get a better decision boundary. Let's not talk about it. And this method was contributed by Jayadeva, who is a professor at IIT Delhi. Well, I'll skip some of it, but sorry, no, it's not I am skipping, it's being skipped. Okay, so we came up with this simple pipeline, which we patented, and then we did a technology transfer, and we are building and validating this product commercially. So it starts with the blood sample. It does a platelet isolation, followed by the PCR, and then we get the gene expression values on which the model is trained to predict the cancer versus non-cancer. It works with 11 genes at the same level of accuracy as 1,000 genes. It's a simple feature selection. There is very little machine learning prowess that I should brag about here. But this kind of ideas came as we talked to many people and we understood the problem clinically, and it made absolute sense to everyone that if you can detect cancer from platelets, amazing, because I don't have any challenge in getting platelets. Then if you're saying that bring 1,000 to, so now here, in the previous case, we showed an application of classification. Here we show an application of feature selection, right? That how you can select features to give you the same prediction accuracy as 1,000 genes or 2,000 genes, okay? Well, the batch effect is being corrected. That's a good catch. The batch effect is being corrected using a more complicated set of methods, which is proprietary in the company, but it basically works with copula, the copula. So that's a joint distribution learning framework, basically. So we are using things like empirical copula to learn the joint distribution, and this empirical method works on the ranks. They don't work. So that's one way to tackle the batch effect. We are not really relying on the values of the gene expression, rather we are relying on their relative expression levels. Okay. I just skip some of it and probably go to the next study. Yeah, so here you have a little bit of the glimpse of what this method does. I'll skip these things. I'll skip some of the slides too. And this is the paper where I reported these 11 genes. What we do is we get to something that we just learned in the previous class, right? That is precision therapy in cancer. So how to define, now we are coming to another definition. Yeah. That's given in the paper itself, and we have patented this also. These are some not so well-known cancer genes. These are not oncogenes or tumor suppressor genes. On the on the contrary, there are some RNA binding proteins, basically, mostly. Five cancers. Okay, okay. Okay, so I know what you are talking about. So they are from, in early stages as well as from the late stages. I don't remember the distribution, but the validation that we did with 250 patients, 40% of them are from early stages, stage 1, stage 2. So we can also detect early cancers, and that's the whole point. One, two, we can detect. I don't remember that, frankly speaking. It could be on either side. It's very hard for me to recall on which side it was more, but yeah, I don't remember that. I'll have to see. I'll tell you what kind of problems might happen. Let's say you have a benign growth, right? So that will have a lot of inflammation in the body. That will have changes in the platelets too. And with that, you can detect, call it a cancer, but it's not cancer actually. Those kind of false positive detections might happen. So we have that chart. It's not, in my memory right now, but it is good. Okay. Uh, I would probably stop with this particular thing, which is AI-guided anti-cancer therapy. Now, if you look at the advent of targeted therapies, patient-specific targeted therapies, there are a lot, small molecules, lot in number. And this is just the past 20 years, and you see a surge in the number of therapies as we go. But on the contrary, the response has not really improved much. So today, in the past about 18, 19 years, from 10% to 30% is the jump in how many patients you can pair with the therapy. You know what I'm saying? So genomics guided therapy. So you have some biomarker in the patient, and in some study, in some clinical trial, you saw that this biomarker, existence of this biomarker is related to activity of this drug in this particular manner. So that means the genomics and drug relation that has grown much, but the response has just grown from 3% to 4.5, 5%. So the response is still bad. Okay. So, and there are various reasons. I don't want to really get into the biology of cancer because when you apply drugs, some cells die, some cells are inherently resistant. They stay quiescent before they bounce back and they cause growth again. And there are a lot of conspiracies going on at epigenetic level and the tumor microenvironment level as well. These are complicated stories. We are not getting into that. We created, again, a definition to this problem, like predicting the right drug for the right patient and how we define this problem. Now, this slide talks about how to define the problem. And the way the problem is defined, you have the gene expression data from the different, because, for training the model, you need training data. So luckily, we have a lot of high throughput genomics, pharmacogenomics data from consortia like CCLE, GDSC, and CPRT, etc. So we can get a plenty of cancer cell line expression profiles and the drug activities. And the drugs you can convert into features, right? You can encapsulate drugs into descriptors. So on one hand, you have the tumor expression profile, and on the other hand, you have the drug descriptors. And of course, you know the response. And these are mainly cell line data. And it so happens that if you apply a deep neural network, it's not that deep, it's a simple feed-forward network with a few hidden layers in between, and you see different ReLU is the activation function. And with that, it's a fully connected network. We give these features, the tumor expression profiles and the drug data, and it predicts whether the drug will work on the patient or not. So this is the model, and that model was built primarily on cell line data. So thousands of 1,000 cancer drugs and 1,000, roughly speaking, 1,000 cell lines, and a subset of the cross product. That's a huge space. And that data was used to train this network. And this network did well, which is reflected in this figure. So it shows the predicted IC50 and the real IC50 and the predicted IC50 and each dot is a pair of drug and a cell line. So you can predict the response pretty well. Then, if you noted carefully here, we considered the descriptors of the drug, not the drug names. So given you give me the SMILE, the configuration of the drug, the molecular details in a vector form, I can predict for the drug too, right? So we did that with two drugs, metformin and orlistat. These two are anti-diabetic drugs, but we applied, and they are finding a lot of applications in prostate cancer. And they have different kind of IC50s, response profiles. So we wanted to see that if we could correctly predict which one among them is more potent. And these drugs have never been seen by our network. Even for a new set of drugs, we can predict their IC50 pretty correctly. And this we also did on mice. Yeah. These drugs were not there. But the drug is being represented as a descriptor. It's a machine learning problem. You see a new bunch of values that defines the drug, and you can predict what is their relative, and that matches with the real concentration. So anyway, we'll skip some of it. It can be used for drug discovery as well, but we are trying to come up with more powerful means of doing that. That's for another day, but our collaborators in Queensland University of Technology in Brisbane did a mouse experiment. So they had this mouse line that mimics metastatic prostate cancer life cycle. So the y-axis, this is a cartoon that shows the y-axis as the tumor burden, and the x-axis is the time progress. So the cancer grows pre-castration, then the castration, then post-castration it reduces, then again it becomes castration resistant, and then you apply drug, then it goes down, and then it goes up and you die, right? That's the typical cycle. So with and this ENZ is the enzalutamide. This is a drug, androgen receptor targeting molecule. Okay, so they experimented with three molecules: enzalutamide, bicalutamide, and apalutamide. All these three molecules are not seen by our model. And we could yet predict the consequence of the application of the drug pretty accurately, which is captured in this. As you can see, roughly speaking, in the initial, we are just looking at the transcriptomics data. And with that, we could predict the drug response. The blue cells indicate sensitivity. The gray cells indicate resistance. And yes, both. Not scRNA-seq. We are taking two kinds of information. The first is the tumor gene expression profile, and the second vector is the drug descriptor, and then we are predicting the response from that. So given that I'm representing a drug as a vector, I can predict for any vector. Well, so that was a mouse study that bolstered our claims, and then finally we used TCGA to show that the patients whose treatments agreed with our predictions did much better as compared to the ones who did not agree with our treatment, our recommendations. This is a Kaplan-Meier. This is the survival curve that shows this blue line are those patients who were given treatments where our model said it will be fine. And the red ones are those where we said no. And you see there's a divergence, which is statistically significant. With that, I would like to stop. It's too much already. So this is the paper that got aired in the end of 2022. it is one of the top 20 health biomedical papers rated by Nature. So yeah, so this is being used actually. We hear from people in our community. Okay, sorry. I forgot to mention that the mouse experiment was contributed by Colleen Nelson. She's a very well-known figure in prostate cancer biology, prostate cancer genomics, and the mouse experiments were done by her group. It's a lot of work. Smriti Chawla is a lab member who went for postdoc at Harvard Medical School. Yeah, not HMI. Now she has come back, joined the company, I guess. So she led this story. For a couple of years, we worked on this. That's it. And I will not even summarize our work, but basically we discussed two aspects of liquid biopsy, CTC and tumor-educated platelets. We also discussed precision therapy using transcriptomics and drug descriptors. We call it a chemotranscriptomics method, right? And AI. So you see, highly interdisciplinary. You have chemoinformatics, bioinformatics, artificial intelligence, microfluidics, at least 5 fields every time. And this is a lot of work by a lot of people, not only us, but we kind of laid it in a way. Well, so I'd stop there with a little bit of food for thought. That's the most exciting thing, which would be very relevant for this audience. We asked, if you have a tumor, like how many of you have heard of natural language processing? How many of you actually know a little bit about it? You know that vector algebra works on natural language embeddings. Like, if you do like, what to say? If you have like, there are funny examples, right? So if you take, let's say, king, right, and you subtract man out of it, and then you add, and when I say subtract, because king is a vector, man is another vector, so vector subtraction, followed by an addition of woman, that will give you queen. That is pretty amazing. 2012 paper, probably by Google. And that was the starting of this area of whatever you are seeing today of GPT, etc., is an output of that. We asked if the same thing can be done with tumors, if you can really subtract a phenotype from another phenotype. So let's say you have, and that is the first time probably we are showing a real application of cancer, application of single cells in cancer. So what we said was, you can take the whole tumor, you can embed it using natural language ways, and then you can subtract every clone, like simple vector subtraction. And you can infer what is going to happen with that. I mean, whether it reduces the risk or not. So basically, that's a more recent work, which I don't think I can talk today. But that makes perfect sense. We saw that in prostate cancer and breast cancer, and we could correctly, like you see here, this is the whole tumor, and you subtract the cluster 2, right? Then what happens? The survival risk drops. This is what you can do with artificial intelligence. You are subtracting a phenotype from another phenotype in an embedding space. And then this one is the most aggressive bunch of cells. You can actually use natural language methods to perform subclonal risk stratification in cancer. That's just for another day. I just wanted to give you a glimpse of that. But with that, I'd stop my talk. And this is a work by Namrata and again, Colleen. Finally, to summarize, we found that application of transcriptomics in conjunction with AI can revolutionize clinical applications in cancer research. And it really does. So two of the works, at least the precision oncology work, where we predict the response of a drug, and the platelets work, we have been able to take it commercially, right? We are working on it. It's not easy. Any biomedical discovery needs a lot of validation. A lot of filters are there before it actually comes out in the market. So we are on that track right now, and that is pretty amazing. This is a work by all the students and a lot of collaborators, and we are working with clinicians to figure out the use cases of what I talked about. I think it's just going back and forth. So these are the people who were instrumental and whom I work with very closely. Some of the students, this doesn't capture all the students, it's the old group. So our lab has graduated like 9 students to date. These are probably those 9 students, not the new ones. Again, we have 10 students who are working on exciting projects. And the collaborators, I basically talked about today's work had Jaydeva, Gaurav, Colleen, and Naveen, and a lot of other folks, okay? So thank you for listening through these talks for 4 hours now. It's too much.